Good afternoon, everyone. We're going to go ahead and get started. I want to thank you all for being here this afternoon. My name is Melissa Ruggiero. I am our Associate Director of Admissions. I am accompanied by our Dean, Dr. Samit Shah. He's the Dean of our School of Pharmacy, and he's the one who's going to be delivering today's webinar on pharmacogenomics, personalizing medicine. Before I go ahead and hand it off to him, I just want to outline a few items on your end. Want to make sure you're all familiar with the chat function. Do utilize that chat function as questions come through. As those come through, Dean Shaw will address those questions. There will also be time at the end for him to address any additional questions you might have. We have gone ahead and muted your microphones as well, so we ask that you use the chat function to get those questions answered or to chime in on any items. And then we will, as I mentioned, leave some time for questions. And then at the very end of the presentation, we'll just be highlighting some information about our Doctor of Pharmacy program that you're welcome to join us for if you find that information to be pertinent to you. So we're really excited to go ahead and get started. And I want to thank you all for being here with us this afternoon. And before I hand it off to him, I just want to make sure the sound quality is good on your end. So if you could just confirm, if I have one of you confirm that you're able to hear me, that would be greatly appreciated utilizing the chat function. Wonderful. Thank you so much, Laura.
Hello, good afternoon and welcome. We are so excited to have you on here today. And I think it really speaks volumes about you to take time out to come and start listening about topics that are relevant to pharmacy as you begin and start on this path. So I'll start a little bit with perfect. I was just trying to figure out the click button. We'll start with talking about the role of pharmacists and what pharmacists do and where pharmacogenomics sort of fits in and why we wanted to do this talk as a potential topic. So we are all familiar with pharmacists working in CVS, Walgreens, the independent pharmacies, in compounding pharmacies, in most of the community settings. But 40% of the pharmacists end up working in hospitals, and within hospitals, they can specialize in cardiology or pediatrics or geriatrics or oncology and so forth. A lot of pharmacists are starting to also work in the pharmaceutical industry, and so there the roles involve taking the drug from the lab to the clinic, so coordinating clinical trials, working with the FDA on regulatory affairs, or working as a medical science liaison, translating the scientific information into clinical language, health outcomes, pharmacoeconomics, pricing of the drugs, and so forth. And then, besides these three major areas that pharmacists work, we are also having lots of new avenues opening up with advances in artificial intelligence, advances in data science, advances in sequencing technology. And one of these is this area of pharmacogenomics. And as pharmacogenomics advances, it's going to create opportunities in two different areas. One is we are going to start seeing community pharmacies and regular independent pharmacies, hospital pharmacies, start offering these services, and it will start becoming a common part of what most pharmacists do. In addition, there will be some specific pharmacogenomics pharmacist roles opening up. And so there's already hospital systems like Geisinger and Kaiser that are starting to have opportunities just for pharmacogenomics pharmacists. And the role of these pharmacists is to set up these kind of services. So with that sort of a background, I want to get into what is pharmacogenomics, and I want to start with a case study here. So in this case study, what you see is these are a couple of different reports. In the first one, a newborn was there, and the mother who had just delivered was given codeine. Now, the mother was given codeine at a very regular sort of dose that's typical for women who have just delivered. And the neonate, the newborn, died after a couple of days. The other one that you see here is codeine was given to a 2-year-old after a routine tonsillectomy, so routine surgery, and they were given codeine at the typical dose that you would give to a 2-year-old. And again, after a couple of days, uh, this child started experiencing some difficulty with breathing. Symptoms quickly got worse, and then the child passed away. And so the question is, for both of these, in both of these cases, we had given the drug at the right dose in each of these two cases, and the physician, the pharmacist, everybody did everything right, and still we saw these adverse effects, these deaths. And the question is, can we go back and figure out what happened here? And more importantly, prevent this from happening in the future. These are just two case studies, but you have thousands of codeine-related deaths of this nature every year, and hundreds of other cases where a patient is not getting the right drug. So can we understand, based on the biology that we understand now, what is happening with these drugs? So, of course, all of you have taken basic biology and you're all familiar with biology, but to make sure we are all on the same page, I'll just start with a little bit of an introduction to basic molecular biology. So we are familiar that all of the cells in the body, or almost all of the cells in the body, except some exceptions like RBCs, contain a copy of the entire genome. So every single cell in the body has the nucleus, and within the nucleus are these 46 chromosomes, which through them contain a copy of the entire DNA. And this DNA, as we know, is 3 billion nucleotides long. Again, most of you are very familiar with this, but an analogy I like to use for explaining this is, think about the genome as a book. And if the genome is a book, then it consists of 46 chapters, 22 of them which are almost very similar, which are your 44 chromosomes, the 1 to 22, but you have two copies of each of them. And then you have an X and a X, or an X and a Y. Then you can imagine all of these sentences in the book, or some of these sentences in each of these chapters as genes, and then each of the letters in the book, that would be your DNA base pair. So essentially, your genome, it's a book of 3 billion alphabets or 3 billion nucleotides. It's composed of 46 chapters with 22 of them very similar to one another, and then an additional X and a Y. Not all of the sentences in the book are very important. All sentences have some meaning, but some sentences are very important, and those are genes. Those are things that would get translated into a protein. So there's certain sentences in some of these chapters which are used to make some information, which would be your protein. And then all of that together is basically your genome. So we have these 3 billion nucleotides, and the question is, so what exactly do these gene and genome do, and how does it impact all of these things that we're talking about here? So we all know about the central dogma of molecular biology, and it's almost ironical that we call it the dogma, because it's based on science, and we really understand this now. And what we understand is that the entire DNA, so the entire DNA, the genome, there are some places that are the genes, around 2% roughly, 25,000, 30,000 approximately genes within the genome. And this message in the DNA is then used to make the mRNA. And then we know, based on the genetic code, 3-letter nucleotides are read by the translational machinery, and they then bring the amino acid corresponding to that particular mRNA, and then we form the proteins. And so the information in the DNA is used to make proteins. Now, the thing is, almost all of us are very identical to one another. 99.9% of our genomes are identical. However, there are some places on the genome. So for example, here we see in the middle, there is a C and a G. Now, there can be cases where instead of a C and a G, some individuals there might have a T, or an A, for example, instead of the C, and a T instead of the G. And so these individuals are going to have something different within the DNA. As you can imagine, if it was instead of a C and a G, it was a A and a T, then instead of the C here in the mRNA, you would end up with an A. And so the amino acid that would then go and get incorporated into the protein would be the one corresponding to an AUA, and it might be a different amino acid. And so in the end, your protein would look a little different. And, and oftentimes these variations have no major consequence. Sometimes these proteins behave almost entirely identical to the normal protein. In some cases, they can be very toxic or have very strong adverse effects. But this is also not very common. So that happens sometimes in mutations that you see in cancer and things of that nature, where a protein's formed that starts messing up the cell. But these are typically things that would not happen in the genetic cells. It's happening more at a somatic level. So the question is, if there are these variations in the genome that are not that harmful, but are just carried over from generation to generation, and if the protein that is produced is slightly different, if it has no effect, then no problem. However, what happens many times is these proteins are what are very important in the activity of the drug. So you can sometimes have proteins that sort of interact with the drug. You can sometimes have proteins that are important in activating the drug. Sometimes you have proteins that are important in destroying the drug and removing it from the body, or metabolizing the drug and getting it removed from the body. Now, you can imagine if sometimes there's a protein with a different amino acid, and that amino acid is right in a place where it's very important in the functioning of the drug, now you're going to end up with a protein that does not work as well. And so the drug activity is going to be impacted because of that difference. So we'll not try to get very in-depth into pharmacogenomics in today's seminar. I just want to give you a very broad overview of this topic. And so we'll just look at one particular activity for the proteins. So the one that we are showing here is that you can have some proteins that play a role in activating a certain drug. So you can have some enzymes in the body. An example is CYP2D6 or CYP2C19. And what these enzymes will do is sometimes we'll give a drug that's inactive by itself. The enzyme works on it, and then it activates the drug. And it's this activated drug that will then go on and perform its activity. Now, what happens in some other cases is that because the enzyme is looking slightly different, remember from the variants that we talked about in the genome, if the gene has a little different amino acid sequence, it might look up different. And if this looks different where it changes its interaction with the drug, then it might not be able to activate the drug in the same manner. And so this individual might not be able to activate and use the drug. So the question is, can we now start using this information to figure out which drugs work in certain ways in different individuals? One thing I want to mention as this goes on, and we'll start getting into a little more in-depth topics here. So please feel free to send on questions to the chat function if there's something that sort of isn't very clear or doesn't make sort of perfect sense. So please do keep sending questions as we go along, and we'll obviously have some time at the end as well. So what are the kind of variations that are possible in the DNA? So we said that 99.9% of the DNA is identical among different humans, but it's the 0.1% or so where we differ from one another. And 0.1% doesn't seem like a lot, but 0.1% of 3 billion nucleotides is a large number. And so there are many places in the genome where we are different from one another. And what are the kind of these differences that would happen between DNA among different individuals? So one difference that is possible is an insertion. So what happens sometimes is the replication machinery is absolutely brilliant, perfect, works perfectly almost all of the times. But every once in a while, it does end up making a mistake. And so what happens is the normal individual might have an A followed by a C and a T followed by a G. But then others might have some extra nucleotide gotten incorporated into that position because of some mistake that happened during replication. And 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 because this is in an enzyme or a protein that's not causing cancer or something like that, this sort of insertion or deletion just keeps living on through the next generation. And it doesn't cause big differences, but it might impact the kind of drug that is being produced. As you can imagine, if you end up having an extra nucleotide, the genetic reading frame gets shifted. And so the protein that would be produced from that point on reads a different language and will come out having a lot of different nucleotides. Another thing that happens is sometimes you can have a copy of an entire gene for some reason during replication, either completely removed when replication occurs, or you might have multiple copies of that gene. So there's a gene called CYP2D6, and it produces a protein called CYP2D6, which is important and binds to a lot of different drugs. And so it's important in activating some drugs and removing some drugs from the body. But what happens is that in some individuals, that CYP2D6 might be just completely absent, or in other individuals, you might have 5, 7, up to 11 to 13 copies of that CYP2D6. Now imagine what would happen if you would have multiple copies of that CYP2D6, for example. You would make tons and tons of CYP2D6 compared to the general population. In individuals who lack that CYP2D6, they are not going to be able to make any CYP2D6 protein. And so drugs that rely on working with the CYP2D6 are not going to be to work as well in these individuals. Another thing that often happens is the variable number tandem repeat. And so this happens in sort of the transcription factor region or the activator region of the gene. So this is outside the coding region of the gene. And sometimes you can have any additional nucleotides. It impacts how the transcriptional machinery binds and starts translation or transcription. And because of that, it can also impact the amount of protein that is made. And the most common kind of changes that we see in the genome are what are known as single nucleotide polymorphisms, or SNPs. And the most common of these is where you would end up having, instead of a C at a certain region, you might have an A. So it's not an insertion or a deletion. You're not adding an extra nucleotide. You're only ending up changing the C to, let's say, an A. So instead of a C and a G at this place on the screen where the red marker is, it ends up having an A and a T. Because of that, you produce a slightly different protein. Because of the redundancy of the genetic code, sometimes the amino acid would be the same, even if the nucleotide is different. So that would be known as a synonymous polymorphism. Sometimes it changes the amino acid, and in some cases, it can result in the incorporation of a stop codon. And so the whole protein translation stops right there in the tracks, and you produce a truncated or a short half protein that doesn't work as well. So these are some examples of the changes we see from individual to individual. And the thing is, just 15 years ago or 20 years ago, we had no way to know what one individual had versus what another individual had, and what these changes were and how we could use that information. But something amazing happened in between 2000 and 2003, and that was that the NIH invested a lot of money into sequencing the human genome. So there are estimates ranging from $1 billion to $2.7 billion. So we spent close to $2.7 billion, and we were able to sequence the first human genome. Now, one human genome is great, but if we want to personalize medicine and if we want to know how each individual is doing and how a medication would work for a different for each individual, we not only need to sequence the human genome for one individual, we need the ability to sequence the human genome for almost everybody. Now, if it's $2.7 billion for one individual, you can imagine for 1,000 individuals, that's going to be an insane amount, and we are not going to be able to do that. But sequencing technology progressed at such an incredible rate that instead that within 15 years, we were able to bring down this cost of sequencing a genome from a few $1 billion to under $1,000. So we can now sequence the genome for less than $1,000. So for less than $1,000, I can give my DNA sample, whether it's from my blood or a saliva or a buccal swab, and scientists can then go in, or a lab can go in and figure out what my entire gene sequence is for less than $1,000. And for pharmacogenomic studies, we don't even need the entire genome. We just need to focus on parts of the genome that are important in producing proteins that would be involved in drug activity. So there are really 20, 30 different genes that we would like to sequence. And so that's an even less expensive test, $200 or $300, easy to conduct. You just simply take a buccal swab or a saliva sample, send it to a lab, and then for $200 or $300, you can get the entire gene sequence for a particular region of your DNA that's important in metabolizing drugs. So I'll give you an example of my test. 5 years back, I went in and I got my genome sequenced. And so now what I have is, when I go in into this particular browser, and just like Google, it's this chromosome browser, I can go in within that, and I can then go in and zoom in on any particular region of my genome. So I know on all my 3 billion nucleotides, which particular nucleotide I have on which chromosome at which place. And I have just randomly here homed in on a particular region on chromosome 10, because this happens to be a gene known as CYP2C19, and it's involved in the metabolism of certain drugs. So that's why I wanted to home in on this particular region and show you what it would feel like. So as you see here, we said that 99.9% of the genome's same from one individual to the other. So on the top, what you see is a reference genome. On the bottom, what you see is my sequence genome. And so on the top, what you see is there's a T, 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 C, T, and even I have a T, 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 C, T, etc. But there's one particular place here where the reference genome and where 80% of the population has a G. But I'm part of that 20% of the population that has an A. And not only do I have an A on one of my chromosomes, it's an A on both of my chromosomes, the one I got from mom and the one I got from dad. And so that's why you see I have both an A and an A on here. And so as we talked, what this is going to do is this individual on the top is going to make a protein CYP2C19 that works well. I'm going to make a protein that's going to be slightly different because of this A instead of a G. Turns out this particular amino acid that's coded right here is very important in the activity of that particular protein. And because I'm having a slightly different protein, my CYP2C19 doesn't work too well. And so there's a drug called Plavix that would just not work for me because of this one difference in the nucleotide here. Now, I want to now go back to the case study that we discussed at the start of our talk, which was about codeine. So we had said here that a mother had taken codeine and she was breastfeeding and the child passed away, a neonate. And then in another case, there was this 2-year-old child, healthy boy, given a normal dose of codeine, and the boy had passed away. So the question is, based on what we now saw about biology and about drugs, can we sort of figure out what had happened in this case? So it turns out codeine is what is called a prodrug. And so a prodrug is something that is mostly inactive by itself, but it needs to get activated into something else more stronger in the body to exhibit its action. And turns out codeine gets activated and turned into morphine once a person takes it. And the protein that is important for converting it is CYP2D6. Now, remember we had talked about all the different kinds of variations that can be present in the genome. In some cases, you can have a SNP where only one nucleotide is different, but in other cases, an entire gene might be missing, or an entire gene, you might have multiple copies of that gene. Turns out with CYP2D6, you can either get a gene deletion, or you can also get gene duplication and multiple copies of that gene. So if individuals were missing the CYP2D6, for example, no codeine would get converted into morphine. And these patients are going to come back to you and say, hey, you gave me the codeine, but it's not having any effect on me. And sometimes they're not lying. What's happening is their body is not able to turn that into morphine. And so another drug might be a better case for these individuals. Imagine what happens when there's a gene duplication. So what happened with both the mother and the 2-year-old child was that they had what was considered a gene duplication. So instead of one copy of CYP2D6, they had 5, 10 copies of CYP2D6. And so their body was producing 5 times the amount of protein that other normal individuals were making. And so imagine what happened then. They took, so in the case of the mother took codeine because she was an ultra rapid metabolizer. She had multiple copies of this CYP2D6. She was making a lot more morphine than the normal population. You know, some of that morphine was, when she was feeding the child, was getting passed on to the baby. And so the baby got an excess of morphine than what they could tolerate. And so even though the doctor did not make a mistake, the pharmacist did not make a mistake, a child passed away. Imagine what happened with the 2-year-old child. They went back and did an autopsy, and what you see here in the case study was this child had multiple copies of CYP2D6, and because of that, this child made a lot more morphine than what the normal child would have made. And because of that, they ended up, again, getting respiratory depression and a lot of the side effects of morphine, and passed away from this incident. But these are examples that I want to share with you as what the potential of pharmacogenomics is. So imagine the future now. You can now conduct a test for $200, and you will now be able to predict whether a certain drug is going to work in an individual or not work in an individual using your knowledge of biology, sequencing, drug action. So it really brings all of these sciences together, and you can use all of this information to help patients and use technology to take drug therapy and medication management to the next level. So what I gave you here is just one example of codeine, and we went through a little faster because it's a 30-minute short talk. But if you start getting into more details, we'll go into much more depth. Here at Regis, we offer an entire elective course on pharmacogenomics, and we go into a lot more detail about what's happening at the protein level, how we dose the drugs, the kind of things that we can do. And these are just some examples of drugs besides codeine: simvastatin, warfarin, clopidogrel, which is Plavix, a lot of cancer therapy, where we can now use this genetic information to guide drug therapy. In fact, there are now 200 FDA drug labels where there is genetic information on how we can, as pharmacists, use the sequencing information to then guide drug therapy. How is this testing done? So there are lots of labs which we can partner with as a pharmacy, whether you're an independent pharmacy working in a hospital. In a hospital setting, you can set up your own labs. In a community pharmacy, we can partner with a lot of these companies such as Genelex or Althea Diagnostics or Myriad Genetics. What the pharmacist will do is they would they can basically take a buccal swab, they collect the DNA, they put it back into this vial that's shown here, ship it to the company runs a sequencing test, gets the sequence for the genes that are involved in drug metabolism, and then they send back a report. And here's a sample report from one of the companies, which would then show simvastatin would not work in this individual, tramadol would not work, codeine would not work, use diazepam with caution, and then examples of drugs that would work, which would be with the green. So the X shows does not work, and then the green shows will work. The warning signal shows this is something that would be based on the judgment of the pharmacist and their understanding of how the drug works and what is going on. So hopefully this gave you a little bit of sort of a trailer of what pharmacogenomics is, what its potential is, and all of the different things that we would be doing as technology advances and bringing it back into our practice. I'll sort of end with sort of a definition of pharmacogenomics. So if somebody is going to ask you, have you heard about pharmacogenomics, or is this something you're interested in, then you just break up this term. And so genomics comes from genes or the whole genome, and pharmaco comes from drug response or how drugs work. And so the term pharmacogenomics is how we use the information in the genome to predict how drugs work, or it is basically the study of the relationship between the differences in the genome and how they impact drugs. And again, what we'll be doing as pharmacists is that based on the sequence of an individual, based on an individual's genetic makeup, we'll be able to say whether this drug should be given at a reduced dose, an increased dose, or if we need to suggest a different medication to a patient based on their genetic information. So with that, I really again want to thank you and applaud you for taking time to start thinking about these topics so early on in your career as a pharmacist. The beauty of pharmacy as a career is that the number of doors and things that it opens up is just limitless. You can work in industry, you can work with data science, you can work in these exciting areas like pharmacogenomics. And our goal at Regis is to support whatever path within pharmacy is the right fit for you, and then give you opportunities for research through dual degree programs, through certificate programs, through electives, to really become leaders within those different areas within pharmacy. So hopefully you enjoyed it. In the future, if there are other topics like the opioid crisis or pharmacist roles within industry, whatever you would like to learn about, please let Melissa or Dan know, and we would definitely try to arrange other things so that you can start thinking about these things as you start your pharmacy career. And so that way you can start building some research or internship opportunities very early on in your company and become leaders in these different areas. So with that, I'll start taking some questions. I already feels that I see some coming in, and we'll start with the first one here. Thank you for the couple of comments. What are some of the ethical ramifications that you see regarding gene databasing, and who owns the information about genetic tests that we administer to patients? How do insurance companies handle this information? Fantastic question, and such a key area. When we offer pharmacogenomics as an elective course in the curriculum, we actually have a couple of lectures just dedicated to the ethical ramifications. The first is, if this gene sequencing information is known to insurance companies, are they going to change the rates for different individuals because somebody has a higher chance of getting cancer, or somebody has a higher chance of drugs not working for them? And this was a big sort of debate. And in 2008, they came up with a new law known as GINA, and it's known as the Genetic Information Non-Discrimination Act. And what it does is it prohibits insurance companies from using genetic information against a patient in determining their rates or in giving them the amount or the rate that they would get. The other thing that is also very common is that many of these genetic tests will sometimes uncover information that the patient does not want to know. So for example, let's say you just did a genetic test to test whether codeine would work for a patient, but you can have incidental finding. And the incidental finding could be that that an individual is more likely to have a chance of getting breast cancer. Is this something that the patient wants to know? They were not wanting to get this test done for that information. There might be implications in the test for their children. What if we find something in the test that's going to be something that might be present in their family and could have consequences for the rest of their family? So absolutely critical questions. And one of a big focus of pharmacogenomics is as this technology advances. In fact, many of you might have heard about CRISPR in your undergraduate curricula or just from looking at science in the field. Not only can we now sequence genetic information, there's a new technology called CRISPR, which is in clinical trials, where you can go in and change. You can go in and change the nucleotide. And so there was a baby that was born in China, very unethical experiment, where they changed their genome so that they would not be able to contract HIV. What are the ethical questions related to that? And so again, a very, very important question, and thank you very much for bringing that up. I'm sorry I didn't really answer that question, but it's a long, another 1 hour talk to sort of think about the pros and cons and the right things to do in many of those areas. The next one is, if I want to learn about pharmacogenomics, do I need to have a PharmD? Pharmacogenomics is something you can learn in your PhD studies. You can learn as a genetic counselor, as a biology major, but most of the current jobs in the area of pharmacogenomics, most of the leaders in the field of pharmacogenomics are pharmacists, because it requires an understanding of biology. It requires an understanding of chemistry and metabolism, drug metabolism. It requires an understanding of pharmacology, how drugs work. And so while there are roles for individuals in pharmacogenomics outside of having a PharmD, for sure, pharmacists are typically the leaders in this area, because the curriculum is just really designed to sort of take this technology and implement that. The next question we have is, this was very interesting. I understand this subject is offered and as an elective course. However, does Regis offer research opportunities? Great question, Sophia. One of the big things for Regis is our really student-focused teaching and research. So all of our faculty, we have small class sizes. Our faculty work with the students in the classroom, and a lot of them have research projects, and they're geared towards helping the students' interest, but also preparing them for residency opportunities. So now there are residencies in areas such as pharmacogenomics or cardiology or oncology, and they're very competitive. And one of the things we do at Regis is we try to have research opportunities during your first or second year, where you can work in the lab with a professor on areas such as immunization or new drug discovery, or a project on pharmacogenomics, or even projects in social and administrative pharmacy, looking at drug outcomes and things of that nature. So we offer lots of research opportunities, and they're all designed to help the student become a better candidate for the job market, for the residency and fellowship market. The next question I have is, what are some reputable companies that PharmDs can work for, intern at, to help develop this field? Great question. There's a company called Assurex. So I know many pharmacists, they work within industry. So there are multiple different roles within pharmacogenomics. One is you can become an MSL, or a medical science liaison. As a medical science liaison, your job would be to convince pharmacists and physicians why this test is important and increase the usage of these tests and help them implement this. So there are companies like Assurex and Myriad Genetics, where there are jobs for pharmacogenomics medical science liaisons. I forget, there's a company on the East Coast, again with the name starting with A, that is also very involved in this field. But again, it could be applied to companies such as Illumina, Althea Diagnostics, Skylos Genetics, where the role is more so interpreting this information and a lead in that area. The other place that you could work with this is in a health system such as Kaiser, Geisinger, Mayo Clinic, University of Florida, Vanderbilt University. All of these are starting to offer pharmacogenomic services, and they want a pharmacist who can take the lead in implementing these services. So in the pharmaceutical industry, in hospitals, and finally, you can take this to the independent pharmacy or to your Rite Aids and Walgreens and start implementing these services even in those settings. So really, it depends on which particular side of pharmacogenomics you would be most interested in, whether it would be the industry application or whether it would be sort of implementing and bringing it to patient care. So depending on that, we can find a place for you to do a rotation or an internship if this is a field that would be of real interest to you. And finally, international pharmacists, definitely opportunities for international pharmacists in this area, as long as you can understand biology, as long as you understand pharmacology and how it impacts. Potential for two different areas as international pharmacists. You can get into research and discovery and keep continuing to discover more drugs where our gene sequence is going to impact how we metabolize these drugs. Alternatively, as a clinician, you can help patients and work on the more patient-facing side of interpreting this technology and helping patients understand this. So these are the questions we have so far. Thank you so much for sticking around for so long. We will, my email address would be available again from Melissa or Dan, and I would be glad to talk more if you'd like to learn more about this. Before ending, Melissa is here and she would just like to wrap this up. So thank you very much.
Dean Shah. Hello, everyone. Hello again. Looks like we still have a good portion of you still here with us today. What I would like to do to just wrap up our webinar, I would like to now transition and just provide a quick overview on Regis University as a whole, talk to you a little bit more about Regis University as well as their School of Pharmacy, if that is a path that you're interested in going down. Now, this information isn't obligatory, so if you do need to take off, not to worry, it should only take about 5 to 10 minutes to kind of just introduce you to Regis University as a whole if you're not already aware of Regis. So Regis University, we are located in Denver, Colorado. We are just north of downtown in the northwest area of Denver. We are a Jesuit institution. So we are one of 27 Jesuit institutions in the United States, and we're one of 2 that have our own School of Pharmacy, the other one being Creighton in Omaha, Nebraska. In being a Jesuit institution, we embody the 500-year-old tradition of academic rigor, intellectual exchange, and faculty commitment to each individual student's success here at Regis, and that includes our students as well as our alumni body. Really a big portion of our Jesuit mission is really our overarching mission of men and women in service to others. And so this is something that transfers over into our School of Pharmacy as well as into many of our other healthcare programs where we are seeking to provide a values-centered education while strengthening commitment to community service. We nurture the life of the mind and the pursuit of truth within an environment conducive to effective teaching, learning, and personal development. So in addition to the clinical rotations our students are going through, they're also taking part in service learning opportunities where they are really exposing themselves to environments and populations different from their own and becoming a more well-rounded healthcare practitioner. So that consists of working with local elementary schools, educating children on health and nutrition, working with the elderly populations, and then later on in the program, students have the opportunity to choose their own sort of passion or area that they want to hone in on. Looking at our pharmacy program as a whole, we are a pretty unique school. We were founded, our School of Pharmacy was founded in 2009, so we just had our 10-year anniversary celebration. In 2009, when we initially started as a School of Pharmacy, we were the only School of Pharmacy in the nation to offer an integrated curriculum paired with a team-based learning model. Since then, a few other schools have adapted our modality. And really what that means is I'll give a quick overview of what team-based learning is all about. And that's really the idea that all of our classes are taught using teams. So that really that core of our curriculum is going to be taught using teams. Students are going to be placed on teams, about 5 to 6 students. Those are their teammates for an entire semester. They get a brand new team each semester. They're doing a lot of individualized reading outside of class or given reading packets that act as the faculty members' lecture notes. They then come to class and they're actively applying what they've learned outside of the class inside the classroom in the form of individual and group assessments, but really the core of those team-based learning classes is what follows those assessments, and that is in the form of group discussions, going through case studies where teams are working with one another to really determine how to best address a problem or any sort of issue in that healthcare field. So it's really honing in on those team-based skills, those collaboration skills, which are so vital to be successful in any sort of healthcare team. That team-based learning goes hand in hand with our integrated curriculum, which is really the idea that students are focusing on one disease or one disease state at a time. In learning about that disease, they're going to learn about the sciences behind that disease, so the chemical and biological processes, and then they're going to also combine that with the clinical and really pharmaceutical care of how to best care for that particular disease. So that's really how that kind of goes all hand in hand. And I do want to highlight our forward-thinking faculty. So with that integrated curriculum, all of our classes are going to have at least 2 faculty members. There's going to be one with their Doctor of Pharmacy degree, so more of an expert on that clinical side, and then one who's going to be more of an expert on the science side. So they might have a PhD in biology, chemistry, pharmaceutical sciences, and they're going to be able to talk more about the sciences behind that disease. In addition to that, teaching really comes first to our faculty members. That's something that they're really passionate about, but research comes second. So we are not a research-funded school, but there are many research opportunities. We have our own pharmacy labs here on campus in which students can work alongside a faculty member in an area of research they might be doing, whether that's through a rotation or just through their own time. So it's a lot of opportunities there. In addition to some of the key components of our curriculum, we're really proud to share with you some of our dual degree options. So really what this means is our students can earn a master's degree while they're working towards their Doctor of Pharmacy degree. If they take the right electives that align up with one of those master's degree programs, they're only taking an added 5 classes. Those will be taken in an online format over the summer semesters. That online format really gives students the flexibility to take a class or 2 over the summer while they might be working, they might be doing an internship, maybe they're traveling. So currently we have 2 dual degree options. Those are a Master's in Health Services Administration, as well as a Master's in Health Informatics. And then we are well underway on working towards getting the final approval for a master's in business administration or an MBA. Alternatively, students may choose they don't want to invest their time in those 5 added classes, so they can do a graduate certificate option. In doing a graduate certificate, students are already taking 3 electives within the pharmacy program that can line up with one of those graduate certificates. They only have to take 1 added class to get that certificate added to their degree. So in that area, we have graduate certificate options in healthcare business management. So that's going to be kind of the shorter version of getting your master's in health service administration. We have one in healthcare quality and patient safety, and then 2 new ones that are well underway of being launched, and that is our grad certificate in data science and then health informatics. So these are not certificate, or to enroll in a dual degree program, you don't need to apply for that. That's something that you would be automatically able to add to your degree program should you be admitted into our Doctor of Pharmacy program, decide you want to start here. So I realize there's still some of you on here. Maybe you are looking into applying into pharmacy programs. We are still accepting applications for our fall 2020 start beginning at the end of August. We are right at that deadline of really that priority application deadline to be considered for admission for fall 2020. That's March 2nd, just a few days away from there. So there is still time to get that application in if you are meeting our requirements. And you would apply through the PharmCast application system. For those of you who aren't familiar with that, PharmCast is a central application system for pharmacy schools. The majority of pharmacy schools are on PharmCast. That allows for you to fill out one centralized application, then opt into the schools you'd like to apply to. You are submitting an additional application fee for each added school, so you do want to be selected in the schools you are choosing. Along with that PharmCast application, embedded in that application, you'll be submitting a personal statement essay. You'll want to make sure you get all of your official transcripts submitted. So you need to submit all official transcripts of all institutions you have ever attended. We also require a minimum of 1 recommendation. And then we no longer are requiring PCAT scores. So if you have taken the PCAT, we will accept those scores, and that can help strengthen your application. But it's simply recommended if you are a little bit on the lower end with a math science or a cumulative GPA. If you're below like a 250, that's typically when we recommend the submission of a PCAT score, but it's entirely up to you. We do a holistic review, so we're looking at all components of your application. So once you do submit your application, we'll then go through a verification process in which PharmCast is making sure everything looks good on their end. They're checking to make sure there's no signs of plagiarism in your essay, checking to make sure your course entry section lines up with your transcripts. They have all transcripts on file. And then once it's been verified, your application would be released to our admissions committee. We'll do our initial review. Should you meet our requirements, then we will go ahead and invite you to an interview, and you'll get to come onto campus and really experience our School of Pharmacy and determine if Regis might be a good fit for you. And those requirements really are our main requirements that we're looking for. Is we put a lot of emphasis on math science GPA. Our recommended minimum of math science GPA is at 2.50. We're looking at your prerequisites. So particularly those science and math classes you've taken in which you need to earn a C- or better, and all of those to start in our program. We also take into consideration if any prior degrees you have, your work experience. We look at your recommendations, and really all of those aspects of your application are taken into consideration as we are reviewing applicants holistically. So that wraps up our presentation for you. I have all my contact details on the screen, so feel free to jot those down. If you would like to connect directly with admissions, you can reach out to me directly. And then if you had questions about today's topic on pharmacogenomics, it seemed to be a really big interest in that topic, I encourage you to follow up with Dean Shah. He's a really great resource for you, and he'd be more than happy to answer any of those questions. So he has left. The presentation is no longer here, but I am available to answer any additional questions you might have. I'm not the expert on the topic that was presented on today, but I can answer any questions you might have about Regis or our School of Pharmacy. What questions do you all have for us? You're very welcome, Sophia. Thank you for being here. Yes, great question. With the interest of time, I didn't have time to go into all the specifics of our pharmacy program, but it is a 4-year program. We consider it a 2+4, where it takes about 2 to 3 years to complete all of your undergraduate prerequisites, to then and then a 4-year doctor of pharmacy program. So students do get summers off following their first and second year. Once they complete their 3rd year, that's when they go into their full-time clinical rotations that begin right after the completion of that spring semester up until they graduate. Yeah, Mahesh, I apologize, I'm mispronouncing your name, but are you wanting to know like all of our master programs or like healthcare specific ones? If I go back to this slide, this is really what outlines like our dual degree programs, which are also offered as individual master's programs. So you can get your master's in health services administration. You can get your master's in health informatics. Some of our other, like, we also have, okay, something related to pharmacogenomics. To my understanding, pharmacy is a graduate program, but it's not a master's program, it's a doctoral program. So just to kind of review like the eligibility requirements for our doctor of pharmacy degrees, you don't need a bachelor's degree. So I don't know where you stand with your educational background, but you don't need a bachelor's degree, you just need those 3 years of prerequisites to be eligible to enroll. But there are options to really specialize in pharmacogenomics if you were to get your doctor of pharmacy degree. I'm seeing you're an international pharmacist. So that tells me that you may want to, I don't know if are you looking to get your license to practice in the United States, or do you still reside internationally? Okay, I see. You may want to connect with us individually. I'm not really sure exactly what you're looking for, but if you reach out to me, I can try to get you in contact with the right person. I honestly don't know if we have the degree you're looking for. I think we have let me take a quick look. Just gonna browse our website really quickly. I do believe we have a so we have a master's degree in biomedical sciences. Are you wanting an online degree? Yes, you're wanting an online degree. Okay, so let me look for you. We do, I believe we have a graduate certificate. I don't know if it's all online. I would encourage you to take a look at our website, go to regis.edu. And I think we have a genetics and genomics degree, and that could be a good fit for you. I'm not sure we have what you're looking for. Wonderful. Thank you so much, Gary. Feel free to reach back out if you are wanting to learn more about the subject matter. You mentioned you're interested in learning about the elective course. Is there a specific elective course you're referring to? Camila, we don't offer a master's in pharmaceutical sciences. Yeah, our degrees are, I would say a lot of our healthcare degrees are gonna be more on the like direct patient care side of things. With your PharmD, there are certainly research opportunities, but I don't know if we have what you're looking for. There are no further questions. I'm going to go ahead and end the meeting. But once again, I want to thank you all for being here. And you do have all of my contact details on the screen. So I'll go ahead and advance the slide there should you need to reach out to me about anything. But thank you, everyone.